This panel suggests literally fixing mistakes within the DNA itself to cure diseases, and it could work. The DNA is an organic chemical of complex molecular structure. It is found in most life cells and even in many viruses. DNA is responsible for coding genetic information and the transmission of inherited traits to future generations. A disease is any harmful deviation from the normal structure or functional state of an organism, often characterized by certain symptoms. It is important to note that not all diseases come from the DNA. Some are born out of bad habits like consuming unhealthy foods or living in unclean environments. I will talk about those too later, but the point is the majority of diseases that people worry about come from the DNA itself that they are born with. The idea is simple. We want to take that DNA, dive in its billion complexities, find the error and fix it. Even though it sounds easy, the real world isn't so simple. First of all, we would need to extract DNA from a bodily fluid like saliva, something which is very accessible today. However, saliva tests mostly analyze only parts of the genome. Clinical grade sequencing, like the kind used for disease diagnosis, often still requires blood samples for higher accuracy, but that's not really an issue. Next, we need to find the genetic problems within that DNA. Technologies like CRISPR are actively working on that. With the human genome being composed of 3 billion DNA letters, we would have to pinpoint the faulty ones causing diseases and fix them. CRISPR works like a molecular search and repair tool. Scientists design a guide RNA that matches the exact DNA sequence they want to edit. This RNA pairs with a protein, directing it to the problem spot. Once there, the protein cuts the DNA, triggering the cell's repair machinery to either delete, replace, or insert new genetic code. This isn't just theory. In 2023, the FDA approved the first CRISPR therapy for sickle cell disease. It edits bone marrow cells to produce healthy hemoglobin, effectively curing the condition. But even with such breakthroughs, challenges remain. Only 5% of diseases are monogenic, meaning they're caused by a single gene. Most diseases involve complex interactions between dozens of genes, lifestyle and environment. Looking through family history can help identify risks, but even then, our DNA repair technology is still limited. Editing trillions of cells in a living body is like fixing typos in a library of books, one by one without mistakes. CRISPR isn't perfect yet. It can accidentally edit their own genes, and delivering it to organs like the brain or heart remains tough. Newer tools like base editing aim to be safer and more precise, but they're still experimental. And like mentioned earlier, many diseases are born from habits and environment, like viruses for example, which can be contagious. Curing those requires a vaccine and a strong immune system. Except from that, seeking a balanced meal, exercising regularly, and getting the right amount of sleep and sunlight are pivotal to the human body. Genetic fixes alone won't save someone who chain smokes or never moves.